Before I start, will people raise their hands if they already know a fair bit about, or a bit about, self-incompatibility? Yeah, that's what I thought. So I was assuming that we'll start with telling you about the biology of self-incompatibility and why one might be interested, and I'm assuming that most people won't know about this. This is a real classic of population genetics. That's why I include it. It's very interesting biology, but it's also a population genetics classic that has a long history, and it's not done yet. It's a topic which has included some juicy and beautiful population genetics and still needs some more, and it's a topic that has a long development of many interesting developments. As soon as we understand one aspect, it seems as if there are more questions that present themselves that we still have to try and understand. So just to tell you what self-incompatibility means, I've defined it in terms of self-incompatible plants. This means plants that reject their own pollen and produce little seed if self-fertilized. And many familiar plants are self-incompatible. If you grow apples, you have to grow cross-compatible varieties because an apple tree won't accept its own pollen. You won't get any apples. You have to have a mate for it. So it's like having separate sexes in the sense that you can't mate with yourself, but it's completely different in that these organisms are hermaphrodites, which you may remember from my first lecture. So they have both male and female function, but the male function and female function don't work with one another. You have to have a different individual, and I'll get to a bit more detail in a minute. That's a little crude. It's actually not that common. If you look at plants, probably there are, the number of families of flowering plants is very unclear. It depends on the fashion among systematists at the time. I think I'm completely out of date and it might be 600, but somewhere several hundred families of flowering plants are recognized, of which no more than about 80 have some species with self-incompatibility. And if you recall my first lecture, I said that outcrossing often breaks down and returns to self-fertilization because of the great advantages of self-fertilization. And so there's no family known to me, or maybe a very small family, but most families that contain several species, if they do contain any self-incompatible species, they also contain plenty that don't have self-incompatibility as well. Now, when I say it's you reject your self-pollen, it's actually not a self-recognition system. If you think of other systems that we call self-recognition, like immune tolerance, for example, this is not a self-recognition where the organism during its development learns some chemical characteristics and learns to accept or not accept itself. There are some organisms where individuals of the same genotype, for example, can't do some function. This is a genetic incompatibility system. So I would make the distinction between self-recognition and a genetic recognition system. It's a chemical recognition system. Plants can't go about and choose their mates and find somebody to outcross with by walking about, as you know, but they are cleverer than you think, and they do these things by chemical means. Plants do many things by chemical means that other organisms do in other ways. In self-incompatibility, pollen is rejected when it comes from a plant that has the same incompatibility type, strictly speaking, as itself, and that means that it has a genetic factor, an allele at the incompatibility locus, sometimes called S locus, that gives it that incompatibility type. And I'll go into a little bit of detail shortly. So self-pollen, but also pollen from other plants that have the same incompatibility type, is rejected, and either rejection of pollen means that it arrives on the stigma surface and it can't grow, or perhaps it starts and goes some way down the transmitting tract towards the egg cells or the ovary, but it doesn't get there. It dies at some point. And these are based on chemical recognition between the pollen incompatibility types and the ovules. So one reason why this is an interesting evolutionary problem is it's a signal recognition system, and several people here have talked to me about working on other signal recognition systems, and some of the properties of those are interesting to explore. So this is an example that may be interesting in the context of this workshop to mention, because there's a great deal of understanding, and because it has a population genetics understanding as well. There are two major types of genetic control, and in both of them, the S locus has many alleles. I'll get to the details of the two major types later. They're in, for example, in the cabbages and broccoli, brassica, within a species of cultivated brassica, more than 90 different S alleles are known. So this is another reason why it's interesting to population geneticists, because up till now, a great many of the talks have talked about situations where we have two alleles at a locus. And we've had hardly any mention, but it should be at least explicitly mentioned that you can have more than two alleles. Of course, it's extremely strange to have this number of functionally different alleles that give different recognition types. But all the same, you shouldn't go away with the idea, if you haven't studied population genetics before, that you only ever have two alleles. This kind of incompatibility that occurs in brassica is called homomorphic incompatibility. It's a purely chemical difference between different genotypes. You can't see what incompatibility type a plant will have, or an apple. You can't tell from the flowers. They look identical regardless of the incompatibility type. And so, as you can then imagine, it's a lot of work to find out the incompatibility types of different individual diploid plants in one of these species. You have to actually try matings, and particularly to determine the alleles, you have to do many cross-pollinations and try and find out who is compatible with who and classify the alleles. But this work has been done, and as I say, in some cases where it's of economic importance, because you want to make hybrids, for example, you want to know that your two parents are compatible, you would have to do the work, and people have done that. Before I leave plant self-incompatibility loci, I should mention that other organisms, apart from plants, do have this kind of system. Fungi, for example, have incompatibility systems that are in many respects quite similar to what I'll be telling you about. I won't give you the details about fungal systems. There are also even some animals, particularly some marine organisms, where the sperm are broadcast into the water, where they have incompatibility systems, presumably to stop their own sperm fertilizing their own eggs. And they have very similar properties, but they're even more difficult to work with than plants. And we know less about them, but we do know that these systems are not confined to plants. I'm going to talk about plants because they're best understood, and the most population genetics has been done with plants. So let me just give you the genetics of one of these systems. This is a homomorphic system. The flowers in this particular plant, this is a species called Enothera organensis, which was one of the classic model systems, if you like to call it that was used for studying incompatibility early on. This is what the flower looks like, and it looks like that, whatever its incompatibility type. And this has a form of incompatibility called gametophytic incompatibility. And you don't need to remember that name. The nice thing about this, the reason I explain it now and the other systems a bit later, is this is very, very simple. As you know, in a diploid organism, so we might have a plant which carries two S alleles, S1 and S2, and its ovules are S1 or S2, and its pollen is S1 or S2, which I've drawn pollen grains much too big up here, being produced by this plant. It will produce 50-50, we hope, S1 and S2 pollen grains. And S1 pollen has the incompatibility type determined by that S1 S allele. And so the rule in this system is that that will be rejected by an S1, S2 plant. It's transmitting tract tissue down which the pollen has to grow, pollen tubes have to grow, will reject pollen that has the type S1. So this S1, this plant, because it has the S1 allele, rejects pollen that carries the S1 allele, and the S2 pollen is similarly rejected because the plant has the S2. So it expresses two recognition phenotypes, and each pollen grain expresses one or the other, and so both kinds are rejected. But pollen coming from a different plant with S3, that pollen is able, you probably can't see this diagram, to grow down and fertilize ovules. But if that pollen from a different plant came from a plant whose genotype was S1, S3, then that S1 pollen is also going to be rejected, and its pollen tubes just grow a little way. So not only self-pollen, but any pollen with the incompatibility type that matches the type expressed by the recipient is rejected. That's why I say it's not a self-recognition, it's a genetically controlled recognition system. I hope that's reasonably clear. That's what the biology is like. And the transmitting tissue can be quite long, and the pollen tubes, the plant has time to recognize this. This is just to show you the length of the transmitting structure in this particular species. This is a sort of record-breaking long one, 6 inches or about 15 centimeters long. Yes? Yes. There's debate about this. A good question. In this Enothera, it is pretty much all or nothing. You the pollen is from the same individuals is pretty much 100% rejected. There's been suggestions by in the past that the genetics that has been done to work out these kind of things. Obviously, if you work with an organism where this is not all or nothing, the genetics is going to be very messy and tough, and maybe the systems that are in the textbooks are those that were easier to work out. And I will come back to something about that later, but there are plenty of systems in the brassica, for example, where it's extremely strong self-incompatibility. It works more or less 100% of the time, although environmental conditions can make it work less well. And so one wonders whether it works completely in the natural population setting, because, for example, if the temperature is high, often the recognition doesn't work quite so well. So there's complications, could be. So before we get to that, can we turn that off for a moment and have lights on the blackboard? Right? We don't need it all away, just the lights on here. So we can have S1, S2, S3, and so on, as I said. So I want you to think about the what will happen in such a population. Now we're going from the basic biology of this system to the population genetics of this system. So suppose a mutant occurs, S4. Will it spread? What will happen to a new type? Yeah, excellent. A new one will spread because if its pollen is compatible on any of the pre-existing ones, it can fertilize any of them, whereas these ones are already in existence in a population. There'll be some numbers of each of them, and S3 pollen will sometimes fall on individuals that express incompatibility type 3 and not be successful. So a new one, because it's rare and not in the population at any appreciable frequency already, will have an advantage. So this is yet another example, you've heard several already, about frequency-dependent selection. It's a rare allele advantage. Right? What equilibrium frequency will we expect after this one has, let's assume that it does indeed spread, it doesn't just get lost by genetic drift. that this population, this allele will come to, or these alleles will come to once we've had this mutation? Different person, please. What do we expect at equilibrium? You have to speak louder, please. Yes, precisely equal frequencies at equilibrium. So, of course, that's making some assumptions. That's assuming that they don't affect anything else except the recognition among one another. But by and large, as far as we can tell, that seems to be the case. So in an infinite population, apart from genetic drift, we'll expect equal frequencies of these. What about the genotype frequencies that we expect in a population? What genotype? These are just allele frequencies. What genotypes will we expect to see? Heterozygotes, yes. All of the individuals will be heterozygotes, because you can only, the pollen can only fertilize somebody who doesn't share that allele in this gametophytic system. And we do see that when people have now identified the loci and have genetic variants that can identify different alleles, you do see that the individuals are always heterozygous. And so that is partly answering Richard's question, do we have some partial incompatibility? Because if we did, we would get some homozygotes, and we don't see that. So we've assumed that all of the alleles are comparable, they're on a par, they're exchangeable completely with one another in all their properties, and of course S5 would spread in the same manner, and so on. And so we could go on and on. We could get, as I said, in Brassica, 90, 100 different alleles. What happens to the selection for S100 if we had S99 already? So we had all of these intermediates, which I'm not going to write up. We had 99 alleles. Think about the 100th allele. Does it have the same advantage as we've thought about for S4? Let's compare those two. It's going to be weaker here, because if you have 99 different ones, you've got all these different genotypes, they can mostly fertilize everybody else already, if they're all at equal frequency. Okay? And this one gets an advantage. Still, you're right. It's good, but to be a new allele, but it's not that much better than the resident alleles in the population. And so at equilibrium, with a large number of alleles, you expect they will all be close to equal frequencies, and the differences between them are not so big. So now think about a finite population. What can happen? Yeah, they're at low frequency individually, and when they get to a lower than normal frequency, it's not that much selection pressure pushing the frequency back up. So it's a stable equilibrium with all these things, but not very strongly so. So the first population genetics that was done in detail on these kind of things thought about the gametophytic system that I've been telling you about. Was done by Sewell Wright in a big and difficult paper, at least very difficult to me, in 1939, where he actually modeled the processes that are going on. So we would have, in his model, a finite population, genetic drift, mutation to new alleles with new types, incompatibility types, and he could predict the numbers of alleles that you would get depending on the population size and the mutation rate, right? And I believe he did this by using this diffusion method, that approach that you've been hearing about, that you can use. And you can see that that could be appropriate for this kind of situation where you've got so many alleles that they're all very comparable in fitness. And he did this for a specific case, this Enothera organensis plant, and what he and the number of alleles that was known at the time was something like 40, or that sort of number, which had all been worked out by doing this difficult genetics, laborious genetics that I mentioned. And what he found was that the mutation rate had to be pretty high, like 10 to the minus 3, given what was known about the population size of this particular plant. It grows in the Organ Mountains in, I think, New Mexico, in a pretty limited region, not found anywhere else on the face of the planet. So there was a puzzle. People motivated by this result, people made efforts to study the mutation rate, which you can do because if you have, say, an S1, S2 plant, you can take a lot of pollen, when you can get very large numbers of pollen grains, and you can see, using its own pollen, whether you do ever get any progeny, and they would have to be mutants to a new S allele, barring contamination. You have to be careful with those experiments, but assuming everything goes right, you can use this to detect new mutations and estimate the mutation rate. And of course, as we would expect now, the mutation rate was not 10 to the minus 3. It was much lower. In fact, as I believe is still true, no mutation to a new incompatibility allele has ever been documented firmly in any experiment. So Wright's conclusion was, well, the population size must be bigger because the population genetics has got to be right. And he was right. Later, it was discovered that there were other pockets in the Organ Mountains of New Mexico of this species. It's highly subdivided, and its effective population size, as Mike Whitlock has been explaining to you, can be much higher than what you would think from counting the number of individuals, and that at least to some degree can reconcile these results. So that was an interesting population genetics analysis that predicted something which later turned out to be correct. Something about the plants that the botanists studying the plants didn't know, and it's always fun when you can do that. And sometimes population geneticists can do that. Okay, so you might think, well, we've solved the problem, we understand this, we don't have to have a high mutation rate, we just have to have a large population size, and we can explain this polymorphism. Yes, question at the back.
So, I'm not sure, like, what is the negative consequence, or the likely negative consequence of self-fertilization? So,
yeah.
For example, if you get a mutation which is self-compatible,
Mm-hmm.
wouldn't, in an equilibrium population, wouldn't that be the biggest?
You have to read my first lecture. And Mike Whitlock also talked about, yes, of course, you're quite right. It's a perfectly good question. Are there negative consequences of self-fertilization? Why will you maintain this? Because a self-fertile mutant could pop up, and unless it's very bad for the progeny, it might spread. It does have a powerful selective advantage, and I explained about that was the main topic of my first lecture. And the notes will be available, the PowerPoint, if anybody wants to see it, with references to who did that work. But yes, you're right. And as I said just now, if you look at the families of flowering plants or other organisms that have this kind of system like fungi, it does break down. You rarely find large taxonomic groups all sharing this. You find large taxonomic groups with a sprinkling of species across the different genera within the Brassicaceae, for example, that have this system, and it is often found to be the same. I'll come to the molecular genetics, and it's often found to be the same molecular genetics within different species in a plant family, but it's different from one plant family to another. Okay, so as I say, this may sound like the end of the story, but actually it's just, in a way, the beginning, because, as I said, this system looks as if you can understand it at one level, and then you start to think about a further level. And the most, to me, interesting paper published on this topic was done by Xavier Wiekemans when he was a postdoc with Monty Slatkin, where they applied the coalescence view of the world, or coalescence approach to doing the population genetics to this kind of system. And so it's interesting to think about self-incompatibility for various reasons. One, for example, is I said that all individuals, well, we agreed, all individuals in the population with this system will be heterozygotes. And a thing I just want an aside to mention is that doesn't mean there's heterozygote advantage. We agreed, in fact, and in fact, if you have 50 or 100 alleles, we know from population genetics actually very unlikely that heterozygote advantage could be the explanation, because in every case, heterozygotes have got to be appropriately higher fitness than homozygotes. It's very difficult to organize such a system with many alleles. Whereas negative frequency-dependent selection, where you have a rare allele advantage, that can easily maintain many alleles. And I just want to, as I say, give a bit of an aside. Many people here, perhaps more people than know about self-incompatibility, may have heard of major histocompatibility loci in animals. Yeah, I'm seeing more nods than I saw hands earlier. Good. In that system, too, we have a very high frequency of heterozygotes, and we have many, many different alleles, and it's possible that heterozygote advantage is operating, but it's also possible that frequency-dependent selection is operating. And to this day, we don't really know what selective force is maintaining the variation, even the amino acid variation, at major histocompatibility alleles. Self-incompatibility is much simpler because the fitness is, if you like, or the reason for maintaining the variation, that's understood. It emerges from the basic biology, as I was saying before about separate sexes and sterility mutations. We don't have to write down fitnesses. They come out of the biology of the system directly. And so when Wieckmans and Slatkin attacked this problem, people had, however, been modeling it thinking of heterozygote advantage for a long time. And even today, let me make a little bit of sermon. People often say heterozygote advantage when they mean balancing selection. And that's absolutely not the same. They're not the same thing. Heterozygote advantage is a force that can produce, it's a kind of balancing selection. It can produce polymorphism of alleles. Frequency-dependent selection, rare allele advantage type of frequency-dependent selection can also maintain different variant alleles. It's a form of balancing selection, and there are other forms, but you should be clear which one you're dealing with in a particular case. But it turned out that to quite a good approximation, you could understand a bit about self-incompatibility polymorphisms, making that approximation that there's heterozygote advantage. And what Wieckmans and Slatkin did was say, no, that's not really what's happening. Let's actually study what the coalescent approach can tell us about the ancestry of these alleles with the actual biology of self-incompatibility, not making an assumption. And what they showed, one thing they showed is that actually this is a form of selection which is stronger than any conceivable heterozygote advantage. If you want to try and compare selection coefficients, no case of heterozygote advantage, however strong the selection coefficient, can give you this polymorphism as you get with rare allele advantage. But what they did do was they worked out the time that you would get till common ancestor of S alleles occurring by mutation under the process that actually occurs. And they compared that with what you would get for a neutral mutation, or neutral mutations occurring in a genealogy. And of course, no surprise, what you find is that the alleles as a whole, the process is much, much slower. That's just a reflection of the fact that you've got the alleles maintained in the population potentially for very long times. When an allele gets rare, selection is promoting an increase in its frequency again. It's also a reflection of the fact that mutations are rare, I told you already. So if you make reasonable assumptions about the mutation rate to new S alleles, which we don't know because we've never done an experiment big enough to even find one of them, or maybe there are other reasons we can't find them, and you work it out, you find a very long coalescence time, hundreds of times older than the neutral case. So maybe we can have the slides on again, and I have that conclusion written. So the allele ages can be very long. We're talking, remember here, about new S allele, new incompatibility type. But the interesting thing, Wickermans and Slatkin showed, it's no surprise that it's long, but it's just like a neutral genealogy in its shape and the relative branch lengths that you expect, but on a much, much longer time scale. And I've given the reference to the paper here. And this predicts even that the alleles could be maintained since before even species split from one another. And this, you can't quite see, it's gone off the bottom of the screen, is called Transpacific polymorphism. And this is, in fact, what you see. So these are some of their results. This shows the equation they came up with, which is in their paper. We needn't go into the details. This is what you get if you assume just that these alleles are maintained by heterozygote advantage or overdominance. And this just shows some numerical calculations for different population sizes. And you can see that it's actually very much older than you would get for overdominance if the population size is small. That's because the small populations don't maintain so many alleles. So if they get close to being lost, the force pushing them back towards the equilibrium of equal frequencies is strongest. If you go to bigger populations, the approximation of using overdominance gets better. But this is what I'm saying, it's stronger selection than overdominance. So this is a complicated figure, but what I'm trying to show you here is that you can have transpacific polymorphism. So here we imagine a species, an ancestral species, that I probably should have labeled an ancestral species. Let's just imagine, look at two S alleles, X and Y, that are in that population, and they will have some differences from one another. This is now just drawing a picture of them in the way we're accustomed to seeing pictures of genes with some exons and introns, and they would, I've colored them different colors to indicate that they encode different incompatibility types, different specificities, and that there are some, these vertical lines are meant to be some of the differences between them. One's in the exons, which I'm imagining that's an exon, the broad bar, for example. There's perhaps an amino acid difference that is the reason why this one is SY, and there are two amino acids that characterize this as an SX incompatibility type. And so these are those variants that have occurred during the evolution of those two types. And then we imagine that you have two species, the species splits into two, and each of them will inherit an X and a Y and many other S alleles, no doubt, but looking at just X and Y. Then within the lineage leading to this species 1 or population 1, if they're very isolated population, you could have another variant that didn't arise before the split, and so I've drawn it on there, but it would occur within SX, and it probably will not be present in the SY allele because this is just a single locus at the moment we're thinking about, and recombination would have to happen for the other allele to get that variant, and that takes a lot of time. And similarly, a variant might occur in the other allele in that species, and similarly, variants might arise in the other species. And so what we'll see is we'll see variants, these ones since the species split, that are unique to one species or the other, just like in any ordinary respectable gene, but we won't have lost the variants that characterize SX versus SY. And those would be shared. If both species retain both S alleles, then we will see those polymorphisms shared between the two species. And that's what we do see. Obviously, if you go far away at another part of the genome, then variants arise within one species or the other and recombine. In this haplotype at some point, the divergence from the SX haplotype will no longer be seen. And this is an example not from a gametophytic system, but it really doesn't matter. It's just that this picture is easier to see the transpacific polymorphism than in some of the other pictures because the colors are clearer. Here we have two species in the genus Arabidopsis in the Brassica family. They have self-incompatibility, Arabidopsis laurata and halleri, two different species, quite close relatives, and they're colored red and blue. This is work from Vincent Castric and his colleagues at the University of Lille in France, and he made this beautiful diagram where you can see the colors, and he also showed you the pairs. So here's a pair of alleles in the two species. They're identical in sequence, and here's a pair next to it of alleles in the two species which are not quite identical. They've got some of those species-specific variants that have arisen in this particular S allele. And you can see lots and lots of pairs, sometimes identical, sometimes with a variant in one species here, but not the other. But lots and lots of pairs, plus the occasional allele that has been found in one species here, the blue species, but its pair hasn't been found, or perhaps it's got lost from the other species. And here's one from the red species, which doesn't have a pair, but many, many cases where there are pairs. So you might even wonder about Arabidopsis thaliana. To get back to the question I was asked earlier, sometimes wouldn't these plants lose the incompatibility and become self-compatible? Yes, Arabidopsis thaliana is a self-compatible, highly inbreeding species. And if you think about it, at the time when it lost incompatibility, it presumably had a mutation somewhere in the pathway that deals with, after this recognition has happened, that does the rejection of pollen that's incompatible. At that time, it presumably was like one of these incompatible species. It must have become self-compatible, but it would not necessarily have lost this variation right off. Those variants now, the mechanism, the machinery to react to the recognition reaction is no longer reacting appropriately. It's accepting the pollen, whatever type it has, but that doesn't mean you lose these alleles. If they started off, as we've understood from the population genetics, at intermediate frequencies, they probably had various different alleles at intermediate frequencies when that loss of incompatibility happened. And we do indeed see three alleles, this one that looks like that, and one that looks like that, in Arabidopsis thaliana populations to this day. So this is transpacific polymorphism across a considerably bigger distance. We can actually recognize within this self-compatible species that's probably been self-compatible for a few million years, many generations. It's a weed that spreads rapidly. We can recognize the ancestral allele from its self-incompatible ancestors. It's got mutations that make it non-functional in Arabidopsis thaliana. And this is a different one that also corresponds to a pair of these transpacific alleles. And again, for those of you who know about major histocompatibility, people have also recognized that there are transpacific polymorphisms, even alleles recognizably very similar between different mammalian species, for example. Okay? So high allele numbers, we know from actually testing them that they have different incompatibility types in these two incompatible species. This polymorphism goes back a long way before these two incompatible species diverged from one another, and even much longer ago than that. Although when we go, for instance, to Brassica, we can't easily see alleles that in Brassica self-incompatibility loci that correspond with these ones. So at some point, things get less clear. So that's all talking about different incompatibility types. What Wekemans and Slatkin also did was they considered the variation within an incompatibility type. So you could have a whole bunch of alleles that have, this particular incompatibility type. They're not, they are labeled here. So we're going to take this one. It's called number 11. You classify them, you give them numbers. And you can take a whole bunch of different plants that have that incompatibility type, that allele, and you could sequence S11 alleles and see what you see. And Wieckmans and Slatkins also worked out, again, thinking of it as a coalescent process, what will you expect among different copies of the same functional type allele. So this is an interesting distinction that we don't often think about. But you can divide things into functionally different alleles and alleles with the same functional type, but those don't necessarily have to have the same sequence as one another. And I can't remember. Why is it not responding? Can't go to the next. Oh, no, I don't want to be that one at all. Sorry. Maybe I'm supposed to be on this page. I think I put my slides slightly out of order. At any rate, what I was going to do is write it on the board, but I see I have it there, so I'm going to just go to that one. So let's think about that. We've got two alleles, just to keep it simple again, allele type 1 and allele type 2, just like I showed you before. And in allele type 1, since it evolved, we sometimes had a mutation at some close by place in the DNA sequence, in the gene or close to it. And that, if it arises in that particular allele type, it could spread through genetic drift, because all of these alleles of the same allele type are equivalent, as I've said. And ultimately, it could recombine onto other allele types, but that takes time. Similarly, the other allele, type 2, might have its own private mutations, which might ultimately happen to spread throughout all allele type 2, but it takes some time. So within a functional category like the red or the black, each of them will have some variation, and it'll be distinct from the other functional type. And so some of these will be neutral variants, synonymous mutations, and they could even be some amino acid differences which don't alter the functional type or make it very bad in some way or make it bad in some way that don't affect the fitness of that allele type. And so what we would expect, and Wieckmans and Slatkin worked out the population genetics, they showed that because you have in S alleles many types, I've just shown 2 here, the effective population size of each functional type is going to be small. If you have 50 alleles, it's going to be very small. So this process here, either these variants that occur in type 1, for example, out of 50, will get lost, or else they'll fix within that type by genetic drift. And so you will have quite low variation, just as you've seen in other contexts where you have a low effective population size. You take a sample of that particular, you could call it a subpopulation from this locus, this species, and you expect low variability. Okay, I hope that fits. Everybody understands that in relation to what you've already heard in the course. And similarly, this one will have low variability, but they could have some fixed differences between them. I've not shown them as fixed here, but they could ultimately be fixed. So we have subdivision within the species. I mentioned subdivision within a species, genetic subdivision. When I talked about X and Y chromosomes, that's a very simple example. The Y chromosome is an isolated population, genetically speaking, from the X, because it doesn't recombine. Here, they're not completely isolated, these alleles, because recombination could occur in this region, but they're isolated because recombination is like migration in a geographically subdivided population. And in this case, if you go to the genome region very close, it's a low frequency that will happen each generation. And so migration is maintaining, for a period of time, differences between them, between the 2 allelic types. Sorry, my symbols have got lost, but I've explained it all. So what you're going to expect is the typical signature that you would see with subdivided populations. You would see, if you didn't know about the subdivision, if you looked, say, in a genome scan and you didn't know that the S locus was being studied among your other loci, you would see high variability because you've got these differences between the subpopulations, if you like. You would see, on the whole, an unexpectedly high frequency of variance at intermediate frequencies, a high proportion of variance at intermediate frequencies, because they would be like this, similar to the ones that actually define the recognition types, and they would be at roughly equal frequencies if the population really had come to that equilibrium that we expect. So those are, as you've already heard, the kind of footprints that identify balancing selection, and indeed we have balancing selection here, but it's interesting to think of this case of balancing selection as a case of subdivision, genetic subdivision within the population. You know, it really is not wanting to go on. So this is just more about some real-life data, empirical data from the polymorphism within one allelic type. So I showed you this picture before, and in this one, each incompatibility type was represented by one sequence. But here I'm pointing to the allele 01, the first one that was found. It can be recognized in both those two species, the blue and the red. And this is data from those two species. Here's Arabidopsis lyrata, 5 copies that all have that incompatibility type were sequenced, and this is just an alignment which just showing, like Brian showed, just the variable positions, and there are synonymous variants at some of those sites and non-synonymous variants at some other sites. This is a larger sample from Arabidopsis halleri, which turned out to be a better system, better organism for working with, because you can actually find the plants and populations in France where Vincent Castric works, but actually this is all work of Xavier Wiekermans, following on from his theoretical work to show that these things are indeed seen when you get empirical data. So it's his lab, and they chose this species, and so they were able to get good samples. But these are all the same incompatibility type, and you do see variation within them. And when you do a 4-gamete test for recombination, which I think has been mentioned earlier, you do see the possibility of suggestion that sometimes there may be recombination, or may have been recombination events in the history, perhaps long history, of this type being maintained in these two species, so since before these two species split. So plenty of time for recombination to have occurred. That's what it actually looks like. So another piece of population genetics that you can do, and Brian did it, is to work out how you expect that signal of subdivision to disappear as you go further away from your S locus, or from the part of it that's maintained by balancing selection. We can see this in terms of the recombination rate. That's what will determine the whether this is a large genome region, like a Y chromosome, that's differentiated from its allelic form, in that case the X chromosome, where there are just two alternatives. This one has many, but it's the same concept. At what genomic scale will you see recombination going away? There had been for a long time in an early, many of the early papers and reviews about self-incompatibility will tell you that this is a non-recombining region. And so we were interested, now it's possible empirically to test that. And you need the theory, the population genetic theory, to tell you what you expect if there's some recombination at a reasonable rate. And we could potentially estimate that rate for these organisms using that kind of data that I just showed you. And I'd never believed that it was a non-recombining region, and that's what the results happily do tell us. If you look, this is just showing the scale you can't see, the x-axis you can't see very well at the bottom, is showing you a picture of the genome region with the S locus that we've talked about is here, and other genes are in the region there. And it doesn't go out for very far on the chromosome. These are the immediately adjacent genes. And we can find those genes because the Arabidopsis thaliana genome has been sequenced. So we can recognize the gene, as I showed you, and we can recognize the genes which are its neighbors. And we found out that they are the neighbors also in these other species. What we see at the S locus here is enormously high sequence nucleotide silent site diversity. That's because we have these many S alleles, each of which has not only the whatever amino acids are required to give you the different specificities, but also these companion variants that have occurred in one allele but not yet crossed over into other alleles. This is a signal of that subdivision increasing the effective population size, for those who like to think that way. And you see that the loci either side don't show nearly such an extreme effect, but still some of them are quite high. Diversity in the rest of the genome, this is Arabidopsis lyrata, is a good deal lower than 10% for silent sites. It's more like 2%. And you can see it seems to be falling away, though, with not enough data here to see a clear pattern. If you look at another measure of subdivision, Mike Whitlock talked a lot about FST. This is just FST applied to sequence data, KST it's called. We do see between the different functional types of S alleles, we see very, very high subdivision, as if there is no migration between them, or very little migration between them. In other words, very little recombination. And that extends to one locus on either side of the S locus itself, but we don't see it for the rest of the genome. And if we look at between population of this plant, FST, the real geographic subdivision, of course, just like Mike Whitlock would lead you to expect, you have the same selection going on for incompatibility alleles in each of the different populations, and so we expect low subdivision when you look between populations. And this is what the theory predicts for FST or KST. It should go down with distance. This is plotted in kilobases using estimates of the likely recombination rate in the region. And it agrees. I didn't put the, I don't think the squares here are the real data, but I can't remember. It does agree very well. You can get a good fit. You can even use it in a way to estimate the recombination rate. So it's all behaving just as the population genetics says. And it's quite a complex situation, quite strange. But it all makes sense. So I've said that, so I got slides out of order, sorry. This is just to come back to that point about differentiation between geographic populations. You have the same kind of selection going on within each population, and so you actually expect less differentiation than you would get for something under balancing selection that's operating similarly in different populations, less than you would get for neutral alleles. And this is just showing that effect for different strengths of selection, and you can get quite, so this is weak selection, NS equals 1 up there, and if you have strong selection, like in self-incompatibility, you could get a lot less subdivision than this is for increasing neutral FST. And this, the population genetics of this was worked out by Mikkel Scherup when he was a postdoc in Brian and my lab. And so quite a lot of phenomena can be explained by the population genetics. And they are in fact seen in empirical data. This is an example of another self-incompatible species in the Brassica family, Leavenworthia species. If you look for neutral genes between populations or the same species of Leavenworthia alabamica, or between two different closely related species, you get, these are the neutral gene FST estimates. If you look at the S locus, it's a lot less differentiated in both cases. So high FST for neutral genes, because these are quite subdivided populations, they only grow on certain ecological contexts and they're separated from one another, they're subdivided, they don't migrate very often. But if you look at the S locus, there's much lower FST. So that's what is predicted, and that's what you see. So that's a lot of population genetics, or selection of population genetics results, and how they play out in real organisms for homomorphic, gametophytic self-incompatibility, the simple kind that I told you about. Now, there's another kind of self-incompatibility that is important. This one, it's called heteromorphic incompatibility. And this is a kind of incompatibility where the flowers are different. I told you in homomorphic, you have just one flower type. You have to do the crosses to determine incompatibility types. In primula and some other plants, it's fairly rare. You have two, or in some cases, three different kinds of plants. One kind, this is called long-styled. I don't know why those two are the wrong way around. This is the stigma, and the pollen is produced by anthers which are held low down. And the other, flower morph, they're called, is the reciprocal. The stigma is down and the anthers producing pollen are up. And you may be able to see these dotted lines. This is telling that the pollen from this type works only on the stigmas of that type. It's self-incompatible, so it doesn't work on its own. And similarly, this pollen is compatible and will fertilize that morph, but not its own morph. This is what they look like in reality. The long-styled is often called pin because the stigma is sticking up. You can see it like a pin in the middle of the flower. And the short-styled looks like this with the anthers showing. You can't see the stigma because it's buried down lower there. Darwin discovered this. The two kinds of flowers were known to children in the village where he lived, but the phenomenon was not known to botanists, and Darwin discovered these relationships by doing the pollinations and discovering the incompatibility types. And this is an interesting case because there are multiple flower differences, as I've explained, between them. It's not just the position of where the anthers are after the flower develops and matures, also where the stigma develops, but the incompatibility types as well. And so it's believed that different genes must control those different developmental differences, but the genetics, which Darwin did himself, and was later done by other people because Darwin didn't know about genetics and didn't realize that he'd discovered Mendelian genetics. The genetics is a simple, single Mendelian genetic locus. So we believe that the different phenotypes are controlled by alleles at least 3 different loci, which segregate together because they're very tightly linked. And we still don't know whether that's true empirically. The molecular genetics has not been done to find these loci and identify them and find if that's true. But we do know that in the homomorphic incompatibility types, that it turns out that in these, unlike everything I've been telling you so far, so please let me say, everybody wake up. I told you it all wrong up till now. I talked about the S alleles and the S locus, and that's true. We use the word locus to mean a single gene in genetics, but we also use it to mean a place in the genome where that gene is located. And in this case, in incompatibility, the ones that have been studied, where the molecular biologists have got their hands on the thing and worked out what's happening, it turns out that there are 2 genes. So in this case, in the brassica incompatibility type of system that occurs in Arabidopsis laerata and Halleri, there's a gene for the female part of the thing, which I did pink for girls, and blue for boys is the pollen gene. In fact, some of them have more than one copy of one or other of these genes. We don't need to worry about the details, but the important thing is that there is a genome region that includes more than one gene, 2 genes in this case, that are involved in incompatibility reactions, and it's not a self-recognition in terms of one locus alleles producing a protein that interacts with itself, which I would call a like-for-like recognition, is what I call a lock and key recognition. Something is produced by the proteins encoded by these two genes, they're different proteins, completely different types of proteins that have to recognize in some way the one from their same haplotype in the genome. And in fact, they behave like a non-recombining region in the sense you can see in this diagram. The size of the genome region can be different between alleles. Again, I've just shown you two, the first two that were characterized by the Nasrallah lab at Cornell, but many more have been characterized by the people in Lille now and the Nasrallah lab as well. And they look as if they're non-recombining. The size of them is different. It's not huge, a few tens of kb. The arrangements can be different. These cross lines show that they can be in different orientations, these genes, and different arrangements. So it may well be that there's a small genome region that doesn't recombine, but it's certainly not anything like a sex chromosome region. Just to give you a case of a gametophytic system, we know that the same thing is true. There's not a single gene. The molecular genetics of the gene that works for the female function is totally different from the one that works in the brassica system, and the pollen gene is also something completely different. The molecular mechanism is different, but it's a recognition system. This is from a cherry species, and a lot more is now known. So that should be a surprise. And there's another complication which I haven't mentioned. In the primula system is what we call a sporophytic incompatibility system. The long-styled form is the homozygous genotype. It produces one kind of pollen, because it's homozygous. And the short-styled form is heterozygous, and it segregates for two different kinds of pollen, but it has one incompatibility reaction. Its incompatibility reaction is such that it accepts pollen from the long-styled form and not the short-styled form. So that's a case of dominance. The short-styled form is dominant, and the long-styled form has the recessive alleles, presumably these several different alleles that control the different developmental systems. And something is understood about the evolution of dominance in this case, but I haven't got time to go into it. So this is a sporophytic system. Here, the pollen from the same plant, it produces, of course, S1 pollen, pollen carrying S1, pollen carrying S2, as far as the allele that they carry. But for some reason, which again is a very interesting story, but not relevant to this workshop, pollen is of type S2. So I've drawn them with the same gray symbol for S2. And the pollen, S2 pollen, is rejected, but pollen from another type with S1, S3, if S3 is dominant, then S1 pollen or S3 pollen both have the S3 type, and so S1 pollen can work even though the plant shares that allele. So that's a little complication. It's called sporophytic self-incompatibility, and that's also seen in the heteromorphic systems. So at equilibrium, when we have dominant and recessive alleles, the different incompatibility types, of course, should end up at equal frequencies. Like in the long-styled and short-styled of primula, you have 50-50. It's very much like having two sexes, except that they're both hermaphrodites and each one can fertilize the other, instead of each one only being able to fertilize the other sex. They can both fertilize each other. So at equilibrium, the types are equally frequent, the two morphs of heterostyle type. But in the systems with sporophytic self-incompatibility with more alleles, some, as I said, are recessive and some are dominant, and recessive alleles obviously would have higher equilibrium frequencies because they can also appear in heterozygotes and not be rejected. And so they come to higher equilibrium frequencies. If you think about the frequencies of all the incompatibility types being equal, the allele frequencies will differ. And so the amount of diversity we saw, I showed you this one already, this particular allele 01 happens to be the most recessive allele, and so it has the largest effective size. We expect it to have the most variability, and if you look, I'm not going to deal with that one. If you look at other alleles, more dominant alleles, you see much less diversity. Unfortunately, at the moment, the sample sizes are smaller for the two more dominant alleles that have been studied. But basically, this table, again, from the work of the allele group, summarizes there are 4 different dominance levels. That, again, may surprise people. Dominance isn't always just dominant or recessive. In this system, you can have intermediate dominance, something which is very recessive, something which is dominant to it, but it's actually recessive to something which is more dominant, and something can be even more dominant still, a different allele. And that's what you find when you do, that makes the genetics even more difficult. That's what you find. So usually the relationships are transitive so that you have a straightforward hierarchy from top dominant to bottom recessive, but even that is not invariably true. So they studied numbers of alleles. Of course, it was easy to find more of the more recessive one because they're commoner in populations. They had quite lengthy sequences. How many haplotypes you found? Many more per for the recessive allele than the others. And if you look at the nucleotide diversity, synonymous site diversity, that one was the highest. And I say the sample is not big enough yet to be sure that this pattern is convincing, but it does go the way you would predict from the population genetics. That's what I was going to talk about, and for some reason I got my slides slightly mixed up. If there's time, I just want to talk about the evolution of self-incompatibility. The main problem that we need to deal with is really this problem that when the molecular biologists worked this stuff out, they found that the thing is, as I said, a lock and key system. It involves 2 parts, a female part, the lock, we could call it, and the pollen. It's a short protein that you might think of as acting as the key. This just shows you the full horror of the details which we have no concern with as a population genetics or evolutionary problem. Right, so I need the lights on the board again, and off there probably. So I talked about mutations before, but now you can see that we really shouldn't have talked that way. I had a piece of chalk. What we should have is the pistil and the pollen. S1 is a haplotype with pistil, allele 1 and pollen allele 1. And S2 is a haplotype, like that. So, and we can have 3 and so on. So how is, I can write 3. Now we're going to look at this one. How are we going to have, what's going to happen if we change the pistil to 4 here? What would be its state at the pollen gene? What could it have here? I mean, we're not going to have 2 mutations happen at once, right? So it's not going to be 4. Are you with me? Could be 1, 2, or 3. So let's say it's 1. Right? So its pollen is just like before. But now, if you look, it's going to accept its own pollen. You know, to reject, you have to have a match. Of course, these are different kinds of proteins. This is just saying what type they are, what allele they reject. So this is going to be self-compatible. So that's not a good way to go. We've not got a new allele. And that's a major puzzle. So the molecular biology, much to my surprise, because I always said has to be a like with like recognition, because otherwise the evolution isn't possible. We can never have all these alleles. We could have this if you had one protein and it somehow has to recognize itself. And then we have a mutation and it's different and it still recognizes itself as different, so that's a new allele. But this doesn't work. This is a real puzzle, and it's not a puzzle that has yet been solved. And so there are ideas about this kind of thing. What could happen? Let me see how to do this. So there's one possible, well, if we start with S1, which is 1, 1, this is from a paper I've given the reference here, which is sort of suggesting what might happen. That you might have, as I mentioned, the alleles that encode, say, the pollen gene or the pistil gene for a particular type are not absolutely uniform. I showed you genetic variability among alleles of a particular incompatibility type, like that 0, 1. They are a heterogeneous collection of very similar sequences, very different from other alleles, but not all the same among the ones which have that specificity. And some of those changes might be amino acid changes that don't damage the function, don't alter it. You're still S1. But maybe you could have a mutation like this that goes to S1. Now I have to get it the right way around. I'm just going to check that I don't do it the wrong way around. I think this is one. Yeah, they call it 1 star. Like that. So this has changed the female side, or this is a variant of the sequence for the female side. So that's what they're drawing in this picture too. They're imagining that there's a sort of wobbly collection of things which they symbolize by a star there. This model that I'm going to show you is in a paper by, or several papers by Marcy Uyanayama from Duke University, who worked out the population genetics of it, and I give a reference and a reading to her paper. This one in Proceedings of the National Academy of Sciences just has the diagram, and some people like diagrams and some people don't, but I have this remark here that this has not yet been modeled to see if it works, and this was now 2004, so 10 years, and I believe it's still not clear whether it works. But I think a version of it, perhaps, that's quite similar, that was published earlier by Marcy Uyanayama, she's done the population genetics and shown how far it works and how far it doesn't work. So I should have perhaps drawn this differently. This is not really a change. We're imagining that this is really a cloud in sequence space. And then we do have a change, so that we've got this one with a change in the pollen. Like that. And it's got to be a change such that now that pollen isn't rejected by all of the S1 stigmas, but only by this new one, or subset, only by things that are characterized by having this star. And of course that's good. That's a good idea for pollen, because now it's not being rejected by as many of the population as before. And we saw right at the beginning, you all helped me with the concept that the pollen, what's advantageous to a new allele is that pollen is able to fertilize more of the population. It's not as restricted in its fertilization opportunities. So this is an example of just that, I hope. Yeah.
But another way of getting used to it is something is actually different molecules or recognizing and matching up.
Mm-hmm.
That molecule itself allows the other pollen and the pistol parts to get accumulate.
Yeah, I'm tempted by such a possibility myself, and I was going to say that sort of at the end, but you'll see these models that don't have any extras, just these two loci, still aren't very easy. This one will work, and Marcy has done the population genetics to show that this one is advantageous, as I've just explained the intuition, and it's correct. It is advantageous, but what it does is, we could call this S2. Now, because it's got just this subset here, and it's got a changed pollen type. So that's a new S allele. It's a funny S allele because you have a reciprocal difference in, if you do the cross between this individuals with this type and that type, one way works and the other way doesn't work, and that's not what we see in real natural populations. So this hasn't got us all the way to a completely distinct S2. And in this one, they have, but they've just done it by drawing a picture, and it's not clear if it works in reality. And so if you look at this, what the population genetics says, yes, this can spread, but actually it replaces S1. So we've got a new S allele, but we haven't got any more than we had before. So at the end of her papers on these topics, Marcy says perfectly reasonably, I think, it's a nice suggestion, that possibly in a geographically subdivided population, a certain population evolves an S2 and it's lost its S1, but other populations retain S1, and so on. You could have different new ones appearing here and there. We have no empirical tests of whether that's really what's happened, and it probably would be difficult to imagine how to test it. We don't even have, at the moment, the population genetics theory to say how much geographic subdivision will work, given mutation rates and so on. Just to complete Marcy's model, though, there's obviously the other possibility is that we start off with a change, or with considering a cloud of variants in the pollen type. This doesn't help us solve the problem, because then you can have another change, so we get like that. But this one doesn't invade, because if a female, if a plant is, if you regard a plant and consider its female reproduction, it's getting it has plenty of compatible pollen. It's not limited by pollen in these models. And there's lots of other genotypes producing pollen that this female is receiving. And here it's no advantage over the other one, or in fact it's disadvantageous, and it will be eliminated. So this one is not going to happen, but that one could happen, and it's possible that it could explain what we have, but it's not easy. There's another model which I didn't put up on the board because I thought I would be running out of time, and I am. People have argued that maybe you can have alleles with dual specificity. They're not S1 or S2, but they have aspects of S1 and S2. They reject both types. And as soon as you start to think about that, you can see that that's not a good thing at all, because you're becoming self-compatible, and you're not rejecting others. Becoming self-compatible is one of the bad things about it. So I won't go into that model. It doesn't seem like a good model at all. I think, so the problem is even worse than I said, because there are incompatibility systems that quite shockingly and amazingly have a female locus, this one here, and multiple linked loci in the same genome region that control the pollen incompatibility types. How this works mechanistically is not fully understood, but there's an extremely nice paper by Tae-Wee Kao's group who discovered this phenomenon, or really nailed it down clearly, and they have some hypotheses about how it might work, which we needn't go into, but of course that makes the problem of the origination of new alleles even more difficult than what I've showed you. And yet here we have a system which is among the record breakers in the whole of biology for how many functionally different alleles you can have. And we can't think of a way by which we could generate new alleles, even though we know they would be advantageous if they arose in populations. So I like Deepa's suggestion that we're thinking all wrong here, probably. Perhaps, and I've always been very tempted by the idea that there's some kind of adapter that works, that looks at the pollen and looks at the stigma protein and says, are you the same incompatibility type? And that somehow, maybe there's a way you could figure out a model to have new ones that involves that. But I've not ever been able to think of one. And it's really quite a difficult problem. There are other difficult population genetics problems that I haven't even touched on in this sort of thing. But this is one of the major things that's been a problem right since Sewell Wright's very first population genetics, because it was always a possibility that it was a lock and key system involving two or more components that had to interact. I think that was really all. Well, I could say we don't understand the evolution of heterostyle. Maybe I've got 5 minutes. I should just mention breakdown because it was raised at the beginning. I made a model of the evolution of heterostyle, but don't let's worry about it. It's another whole story and might well be wrong. And we really need to understand the molecular genetics before we can test it. But heterostyle has been very important in studying the breakdown of self-incompatibility. And this just illustrates the sort of advantages that a homostyle would have. The transmission advantage that I discussed not above, but in my first lecture, it should say, and reproductive assurance. If pollination is not adequate, it would be good to have perhaps a recombinant between those three different loci that I mentioned, could produce a type which has the female parts from one of the morphs and the incompatibility appropriate, and the anther pollen incompatibility of one of the others. They're all present in a heterozygote. A recombinant could produce an intermediate or a recombinant type that would be perfectly self-compatible right off. And that's, it doesn't advance. That's what has been seen in a real population, and Fisher was the first person to study these. This is what's called a homostyle. It looks like a recombinant between those two types. And indeed, the incompatibility is such that the pollen works fine on this stigma. It's close by, and they're quite highly self-fertilizing in the natural populations where they occur. And Fisher's model that I told you about for the transmission advantage of a self-fertilizing variant was used to explain this by Crosby. What Crosby's paper here is about is the fact that the homostyle form would certainly invade and reach a high frequency, but it seems as if perhaps it's not spreading through the whole of this particular species. It may be that it's just, we haven't watched it long enough, like in the experimental evolution, to see what happens in the end. Maybe it will get there. But it certainly has got there in some Primula species. This is Primula scotica. This species is entirely homostyle. And of course that makes sense, because Scotland is a place that insects don't do very well, and pollination is certainly not very well assured. It grows in the north of Scotland and in the islands where it's rainy and very windy. So the continued presence of the two morphs in the other species is interesting, and it would be nice to understand more about it, but it's no surprise that many Primula species have lost incompatibility and lost those floral differences. They're all one morph that looks like a recombinant type. And I think I'll stop at that point. Actually, I would just like to say one further thing, which is going back to the major histocompatibility systems. So as I said, that's another system with high polymorphism, a lot of alleles, and in some way, possibly involved with recognition systems, so some similarity with self-incompatibility. But the way it's studied and the way it behaves have some important differences. One important difference is that recombination probably occurs. And so we don't have a little patch of genome over those loci, like we seem to have with the self-incompatibility that stops variants moving between them. So we probably have to appeal to some kind of selection for explaining the linkage disequilibrium. The associations with certain combinations of variants, sequence variants, are found and others are not found. That's still far from being understood. And because there's recombination, you can't make a phylogenetic tree of these alleles like I showed you, where I showed you those circular trees with the Transpacific polymorphism and so on. That's only valid where you're not having any recombination, a tree. And so for MHC, you can make trees, and people show pictures in their papers of what look like phylogenetic trees, but they're a recombining genome region. And for MHC systems, we don't know the functional types. We know that they have differences, but we can't assign a sequence to a functional type, whereas with self-incompatibility systems, we can say it's an S1, and we can do the tests to show that it's different from an S2 or an S3. In MHC, it's a much more difficult problem to understand the maintenance of that variation for these reasons, and in some ways, it's way behind. But I think the clarification of understanding self-incompatibility, because it is a bit simpler, should help advance understanding in that more complex system. So that really is all I have to say, unless there are any questions. Brian has a comment. Again. I don't know whether it was selected for in the homomorphic incompatibility systems. It's obviously a recombinant, as I've explained in this case of heterostyle, but the same sort of thing would apply in homomorphic system. If you had a recombinant between the component loci, so you've got a locus controlling the female incompatibility type and the male incompatibility type. If you had a recombinant between a couple of these, of course, that would be self-compatible again. And if the ecological situation and the inbreeding depression are such that incompatibility is favored and self-fertilization is disfavored, that would be a bad allele, it would be eliminated. So you can see that it would be good not to have recombination, but that doesn't mean that selection in the homomorphic systems has resulted in the small region where recombination appears not to occur, because these things have been evolving for a very, very long time, and there are rearrangements, as I showed you, and size differences between the different alleles, and it's not entirely clear to me whether selection promoted a lack of recombination and those changes then were able to evolve, or whether the evolution of new incompatibility alleles happened to carry with it some of these differences that stop them recombining with pre-existing alleles. I don't know if you think that's reasonable, but I don't think we know that selection promoted lack of recombination in these cases, whereas I think in the case of Primula, we do expect that the different developmental changes are controlled by probably different genes that probably have been under selection to keep them together and avoid becoming homostyle, although that does happen if the ecological situation promotes it. I hope that's sort of reasonably clear. It's a difficult thing to test whether selection promoted recombination suppression. It's a difficult thing to test whether selection promoted any ecological or natural adaptation, of course. Yes, question there.
I guess it's a bit of a long question, but so when antibodies are produced, at least in the human immune system, you essentially have this sort of VEJ recombination which produces like multiple. So do you have something similar with pollen and plants, where you can create sort of like multiple keys?
No, apparently not. Apparently these are just allelic differences. There's nothing like that. No, no.
But I can imagine that having a huge advantage because
Yeah.
just by pure sampling, you're more likely to produce a key that.
Yeah, I mean, a true self-incompatibility would be the best, where you only reject yourself. So where you learn during your development what yourself is like at a locus or some system, a mechanism, to assay yourself and reject yourself. But of course, these things don't do that. I haven't talked at all about how you go from one allele to two. If you might have noticed, I talked about how if you've got several, how you can get one more. It's even worse to think about how if you start with none, you can get one in the first place, and then 1 to 2 is not so bad and so on, but it's still, we don't have an answer. So yes, if you had a thing that didn't involve any of this, plants apparently aren't that clever. It's not a self-recognition system, it's a genetically controlled incompatibility. And you can't easily say, why does an organism evolve something by one route rather than another often? I mean, what I could say is plants do have recognition systems, many of them. They recognize many pathogens. They have many recognition systems. Possibly these things, a very attractive, tempting idea, is that self-incompatibility, recognizing pollen types, evolved from a mechanism that plants already had to recognize pathogens. And indeed, some of the machinery, the molecular details, which I didn't bother you with, are, things that could be used in, and are similar proteins to things that are used in pathogen recognition. So it's not totally out of the blue that this has to evolve. Plants do have some recognition systems that may have evolved into these, but still, there's big gaps, as you see, in what we understand.